fate mapping studies, it's been shown that during embryonic development, myeloid progenitor cells from the yolk sac are able to see the developing myocardial tissue, and which arise to these tissue-resident macrophages. These tissue-resident macrophages are able to persist into adulthood through self-renewal mechanisms. After birth, however, hematopoiesis shifts into the bone marrow and spleen, and this leads to the production of circulating monocytes. And so as we age, these monocytes begin to infiltrate the myocardium and give rise to this distinct population that has been identified as being MHC2 high. Furthermore, as we age, our tissue-resident macrophages are exposed to inflammatory stimuli that also drives a portion of them to have this MHC2 high phenotype. And so within the healthy myocardium, we have these distinct macrophage populations defined by their ontogeny. And so many studies have shown after myocardial infarction that these macrophage subsets are each subjected to the stimuli of apoptotic cells. But one of the major questions is, how does this stimuli affect not macrophages as a whole, but specifically the heterogeneity macrophage reprogramming of these macrophages? And so the process of removing apoptotic cells has been really well studied in both macrophage populations. And just as a kind of a 1,000-foot view of the process of efferocytosis and how it contributes to macrophage reprogramming, is that upon eating, there's a transcriptional response that involves increasing actin cytoskeleton remodelers, as well as increasing cytokines important for inflammation resolution, as well as lipid pro-resolving mediators as well. And these are important for carrying out the reparative processes induced by efferocytosis. Furthermore, there's an inhibition of inflammasome activation and the expression of inflammatory cytokine, thus once again polarizing these macrophages to an inflammation resolution phenotype. However, most of these studies have just focused on both populations of macrophages. And as I've shown in the myocardium, we have a wide spectrum of macrophages that are heterogeneous, both in their phenotype and their function. And so it's not just ontogeny, but also the tissue microenvironment and previous stimulation that drives the macrophage phenotype along the spectrum. And this leads to a wide range of efficiencies at certain macrophage functions, such as efferocytosis and cytokine production. And so we wanted to kind of take a step back and just look at how do single macrophages display a unique transcriptional response after efferocytosis. And so in collaboration with Dr. Jenny Lin here at Northwestern, we designed this single cell sequencing experiment to interrogate this question. And so for this experiment, I isolated peritoneal immune cells using a peritoneal lavage of mice and plated them ex vivo. And I performed an efferocytosis assay with apoptotic Jurkat cells, which are of human origin. And so having these two cells from two different organisms allows me to be able to identify mRNA transcripts from the immune cell and potential mRNA transcripts from the apoptotic cell that are passenger mRNAs that get incorporated into the immune cell's transcriptome. And so after 6 hours of efferocytosis, I collected my cells and performed single cell sequencing. And so through this process, we were able to sequence over 8,000 cells across all of our conditions. And so the first step I took was to identify what immune cells I was able to extract from my peritoneal lavages. And so using single R, I was able to compare the transcriptome of each individual cell to a reference genome in the ImGen database. And this allows me to unbiasedly identify the immune cells within my sequencing data. And as you can see, I have a large population of macrophages, a large population of B cells, and a small population of T cells, which is pretty representative of what we would have expected in the peritoneal cavity. And so looking closely at the macrophages, I was able to identify 4 transcriptionally distinct populations. As you can see here, by plotting their 50 most differentially expressed genes across each cluster within this heat map, these clusters represent transcriptionally distinct populations. And so with these populations that I identified, I wanted to take a step back and look at how does this relate to what's known about the peritoneal cavity and the macrophage compartment within this cavity. And so similar to the heart, within the peritoneal cavity, there are 2 main subsets of macrophages. Your embryonically derived macrophages that self-renew, termed large peritoneal macrophages, or LPMs. And then you have your monocyte-derived macrophages, termed your small peritoneal macrophages, or SPMs. So through flow cytometric studies, markers for each of these populations has been identified. And so I just looked at the expression of these markers within my data set. So specifically looking at CD11B and F480, which are markers for large peritoneal macrophages, and also CD11C and MHC2, which are markers for small peritoneal macrophages, I was able to see this distinct separation of these populations, and the clustering also correlated with this separation. What was interesting is when I looked at receptors, specific phagocytic receptors such as TIMD4 and MERTK, and also a transcription factor that's important for the upregulation of efferocytic machinery, KLF2, I see an enrichment in specifically this F480 positive MHC2 low large peritoneal macrophage cluster, suggesting that these macrophages may be more primed for efferocytosis compared to the other populations. So furthermore, I wanted to interrogate other macrophage differentiation factors and inflammatory genes to further understand the biology behind this clustering. And so looking at specific genes expression, such as like GATA6, which is important for the differentiation and development and maintenance of this large peritoneal macrophage compartment, I see an enrichment of these blue within this blue subpopulation. Where conversely, looking at gene expression of IRF4, which is important for the differentiation from monocyte to macrophage, I see an enrichment in my small peritoneal macrophages, lining up with previous studies. What was interesting is when I looked at inflammatory gene expression, such as IL-1 beta and IL-6, I see enrichment within this red population, suggesting that these macrophages, either in vivo or ex vivo, were previously exposed to some sort of inflammatory stimuli. And so it was nice looking at specific gene expression and marker expression, but I wanted to look at the 1,000-foot view of these clusterings actually represent different biological functions within these subclusters. And so to do this, I determined the differentially expressed genes within between my different clusters and performed a gene ontology analysis to identify specific pathways that are upregulated within each cluster. And so not surprisingly, in my IL-1 beta producing large peritoneal macrophage cluster, I see a huge enrichment for inflammatory pathways. What was more interesting is when I looked at this MHC2 intermediate population compared to my MHC2 low population, I see an enrichment of homeostatic pathways such as wound healing within this MHC2 low population, whereas I see an enrichment of pathways such as the phagosome and actin cytoskeleton organization, which I would assume is a more efferocytic transcriptional signature, was enriched in these intermediate macrophages, suggesting that these macrophages have already eaten or are efferocytic, performed efferocytosis. And so furthermore, I wanted to look at how my small peritoneal macrophages were different than my F480 MHC2 low large peritoneal macrophages. And not surprisingly, once again, I see an enrichment of these development and homeostatic pathways within my large peritoneal macrophage population. Conversely, in my small peritoneal macrophage population, I see enrichment of pathways that I would infer, such as cell migration. But what was interesting is we saw an enrichment of pathways associated with oxidative phosphorylation. And looking at specific genes within the electron transport chain for different subunits, such as these ATP synthase subunits, you do see an enrichment of these genes within these macrophages, suggesting that just at steady state, these small peritoneal macrophages are more metabolically active than their large peritoneal macrophage counterparts. And why this was kind of really stuck out to us was that it's been emerging that macrophage metabolism is tightly linked to its function, its immune functions, and that these represent potential therapeutic targets that may be important for cardiac repair. And so just looking at the field of immunometabolism, the 1,000-foot view, it's been shown that in classically activated macrophages stimulated with LPS, Luke O'Neill and the Pierces have shown that metabolic pathways such as glycolysis and the pentose phosphate pathway are enriched. And this leads to the expression of HIF-1 alpha target genes, which drives inflammasome production and the production of inflammatory cytokines. Where on the other hand, alternatively activated macrophages stimulated with IL-4 increase pathways such as the fatty acid oxidation pathway and oxidative phosphorylation. And this drives expression of the mannose receptor, a canonical marker of alternative activation. And so with the tight knit function, with the tight relationship between metabolism and immune function, many of these different metabolic inhibitors are showing promise as therapeutic opportunities, as they can drive different metabolic processes, which may be able to improve our ability to enhance or decrease inflammation in certain contexts. Furthermore, our lab has shown that apoptotic cell catabolism induces different metabolic programs within the macrophage. And so Dr. Shuang, a previous graduate student in Ed's lab, who's now a postdoctoral fellow at Harvard, she showed that upon efferocytosis, the macrophage increases genes for fatty acid oxidation, and in turn, an increase in the electron transport chain. And this drives expression of IL-10, a really important mediator of inflammation resolution that enhances cardiac repair. And through her studies, she looked at these bulk macrophage populations. And so I really wanted to look at how the metabolism of a single macrophage changes within my data set. And so as I previously mentioned, I performed this efferocytosis assay with these two cells from two distinct organisms. And so this allows us for when we're performing the single cell sequencing analysis to identify reads that came from the apoptotic cell as they would align to the human genome versus reads that align to the mouse genome, which would be our macrophage genes. And so through this analysis, we see that after 6 hours, pretty much all of the passenger mRNAs from the apoptotic body are degraded, suggesting that at 6 hours, our transcriptional data is free of this potential contamination. And so I performed a pseudotime trajectory analysis. And I think this is one of the coolest and I think powerful things that single cell sequencing data is able to give us. And what this analysis exploits is that biological processes such as efferocytosis occur in an asynchronous nature. And so what that means is that although I fed my cells all at the same time, it doesn't mean that they're all starting the process of efferocytosis at the same time. So when I collect my cells, I have some cell macrophages that haven't eaten. I have some that took their time and just started eating. I have some that are probably still in the process of eating. And then I have some that have already finished eating. And so when you collect these cells for single cell and then cluster them, you get your macrophage cluster, but you lose the physical time variable for your gene expression. And so what this algorithm does is it statistically infers the ordering of these macrophages across a new variable called pseudotime, which creates this trajectory. And within this, you're able to determine the expression of the gene through each of these cells. And so this gives you here a plot of your gene expression as a function of pseudotime. Thus, this is a really powerful way to understand the dynamic changes of gene expression for a specific biological process. And so performing this, this algorithm on my data set, I, not surprisingly, came up with two different trajectories. One that was for my large peritoneal macrophage population and one for my small peritoneal macrophage population. This suggests that even though if a small peritoneal macrophage is performing efferocytosis, it doesn't become more LPM-like and cluster within here. And when I look at specific gene expression that I would expect to be dynamic throughout efferocytosis, such as the expression of ACTB, a actin cytoskeleton remodeler, and MHC2 allele important for antigen presentation, I see an increase of these genes, which is to be expected. However, when I look at inflammatory expression of IL-1 beta, I see a dramatic decrease after efferocytosis, thus suggesting that my pseudotime model is accurately modeling the process of efferocytosis in both populations. And so with this, I was able to determine genes that are differentially expressed as a function of the pseudotime. And so this just gives you genes that are basically have some type of dynamic change through this process. And one of the pathways that was enriched of these dynamically changing genes was fatty acid metabolism. And when you look at enzymes important such as this lipoprotein lipase, which is important for creating free fatty acids, as well as scavenger receptors that transport these free fatty acids into the macrophage, and other fatty acid binding proteins important for creating acetyl-CoA, which drives fatty acid oxidation. I see an increase overall in both my large peritoneal macrophages and my small peritoneal macrophages, suggesting that the process of fatty acid metabolism is a conserved metabolic programming across my macrophage populations. However, when I look at other metabolic pathways, such as glutathione production and metabolism, I do not see this. I see a more distinct expression changes within my two populations. So specifically in my large peritoneal macrophages, I don't see a large increase, although I do see dynamic changes of enzymes associated with glutathione metabolism. However, in my small peritoneal macrophage population, I see a dramatic increase of genes related to the production of glutathione. And what this suggests to me is that at steady state, these small peritoneal macrophages were already metabolically active and therefore probably producing reactive oxygen species more than their large peritoneal macrophage counterparts. And so when exposed to this stimulus of an apoptotic cell, they quickly have to, which increases more reactive oxygen species, they have to quickly find a counterbalance through glutathione to mediate cellular redox homeostasis. And so just to sum up what my single cell sequencing studies have concluded up to this point is that I have these two distinct populations undergoing efferocytosis. And within these populations, I see a conserved increase in fatty acid metabolism, but I see a differentially regulated glutathione metabolism between my large peritoneal macrophages and small peritoneal macrophages. And so I hope these studies highlight the depth that you can go into with single cell RNA sequencing. But one of the big caveats is that we're just looking at mRNA expression, just the transcriptome. And it would be nice to be able to tie this to some type of functional output. And so luckily, Shuang, before she left, performed this metabolomics assay where she had a bunch of her different groups that she was interested in. But I lucked out in that she had two conditions where she had wild-type peritoneal macrophages and peritoneal macrophages co-cultivated with apoptotic cells that she did perform this analysis. And so I was able to take her metabolomic data from these two conditions here and determine differentially expressed metabolites. And then I can take my differentially expressed genes from my single cell RNA sequencing studies, and using this program called ShinyGAM, developed by Max Artemov's lab, I was able to integrate both of these data sets to form a distinct metabolic network that occurs after efferocytosis. And so not surprisingly, a large portion of this metabolic network is associated with fatty acid metabolism. I also see an enrichment of glutathione metabolism as well, further validating my single cell RNA-seq findings. I also see the enrichment of the TCA cycle as well as other metabolic pathways. And so just to kind of jump in and look at one of these pathways a little bit more closely, I picked glutathione as that was one that was differentially regulated within my single cell data set. And interestingly, in Shuang's data set, she also sees a significant increase within glutathione production and metabolism, further quantified here. And so kind of putting everything together into my working model is that through these single cell sequencing and metabolomic data, I was able to uncover a heterogeneous immunometabolic function where glutathione production is distinctly regulated after efferocytosis within these monocyte-derived small peritoneal macrophages. So moving forward, a lot of the big questions that future investigations are aimed at answering is one, does this different metabolic reprogram translate to cardiac macrophages, particularly after myocardial infarction injury? Do I see that these a conserved increase in this fatty acid metabolism, and do I see distinct changes within glutathione production of cardiac macrophages? Furthermore, there's been a close connection of metabolism with the potential epigenetic reprogramming and thus transcriptional programming of macrophages. So that's another really interesting, exciting idea, is that by changing the macrophage metabolism within the heart, we're able to reprogram the macrophage later in life. And so with that, I just want to have a big acknowledgement for the Thorpe Laboratory. Everyone's been super great, and it's a great community to work and be a graduate student. I specifically want to thank Ed for being a great mentor. He's always pushing me to think deeper, think more mechanistically, and allowing me the opportunity to go and present research in forums like this. I also want to thank Shuang a lot for a lot of the data and kind of establishing this immunometabolism arm within our lab. And also want to give a big thanks to Jenny Lin. She's been huge in helping us establish our single cell sequencing pipeline within the lab and at Northwestern. And lastly, the funding sources that have been able to fund me and also fund this research. And with that, I'll take any questions.
Okay, that was awesome. Thank you, Connor, and thank you, Ed, for agreeing to give us that presentation today. So I'll ask anybody who's interested in asking Connor questions to raise their hand via the participant window. And while we're waiting for that, maybe Connor, I can start. So I noticed that in I really like that description of sort of the temporal, process of efferocytosis and how dynamic it is. But I also wonder if you're planning on looking at the temporal apoptotic cell, and does that sort of indicate? I mean, I know that you treat all your apoptotic cells at the same time and you feed them at the same time, but have you looked to see whether or not there are expression levels of phosphatidylserine or, any other ligands for phagocytosis change, and if that affects, obviously, the way the efferocyte?
Yeah, that's a really interesting question. I've definitely been more focused on the phagocyte side of things, but it's especially, I think that's a really important question, as the apoptotic cells, right, are also being in culture for 6 hours. So yeah, if there are changes with expression of different efferocytic signals, I think, yeah, that's a really interesting question.
Yeah. Complicates your interpretation about 10,000-fold, but I mean,
that's what I was thinking. I was like, oh, but
I don't have to hire someone else to do that part. Okay.
Yeah.
Yeah, Natalie, go ahead. I'll unmute you.
Hi,
go ahead, Natalie.
This very well presented, cool data. I really like the pseudotime idea and how it was developed. Earlier in the presentation, when you compared F480 macrophage transcriptomics to F480 negative, and then you went on to validate certain genes, the distribution of, for example, ATP5E, COX17 between these two macrophage populations were widely different. It was a multiphasic distribution in the F480 negative macrophages, and it was bimodal in the F480 positive. So do you think pseudotime plays a role, or you're capturing further subpopulation of macrophages within that F480 positive negative categorization?
Yeah, so I think there needs to be a validation, but I think that's one of the really cool things about doing single cell sequencing and the pseudotime is I do think you're able to actually elucidate these phasic patterns of gene expression, which, yeah, so at least from the data I've collected and this algorithm, I would say that there most likely is phasic process metabolic programming, I guess, throughout efferocytosis, as I can assume when you first engulfing apoptotic cell, you probably have a lot of lipids that you need to deal with to break down that membrane. But then, now you're exposing potentially RNAs and stuff, and now you have to do kind of RNA metabolism. And so I think all of that drives probably this phasic nature of metabolism throughout efferocytosis.
Great. Okay, thanks. Okay, Colin, you're next. Oh, I thought I unmuted you. There you go.
Hey, Connor. Yeah, this is Colin Evans at Northwestern. Yeah, it was remarkable to me how dynamic the changes in the cells were over time. And so I was guessing that in terms of selecting them for, say, potency or how hungry they are, you could choose them by their marker expression. But I was wondering, if you were to ever try to use them therapeutically, would there be any way of keeping them at the level of potency that you want them, particularly if they're in the circulation in vivo?
Wow, that's a really interesting question. I guess if I were to think about using these therapeutically, I probably wouldn't focus so much on feeding or like producing these efferocytic cells, and more focus on, okay, so we see that efferocytosis reprograms glutathione metabolism. And so identifying therapeutic targets within the glutathione pathway, I think that's probably a better route for therapeutics than necessarily the cellular, using macrophages as a cellular therapeutic candidate. So yeah, I think identifying how efferocytosis may be programming macrophages for cardiac repair through metabolism, and then determining therapeutic targets based off that metabolism is, in my opinion, at least, a good, a promising therapeutic potential.
Sure, thank you.
Great. Okay, and our next question is from Henri. Go ahead.
Hi, Connor and Ed. Really exciting work. Congratulations. My question is kind of related to what Katie has mentioned. The fact that some of the macrophages are the first responder while the others are a little slower. Do you think it's like a random process, or those first responders are programmed to be the first responder?
Great question.
Yeah, that's a really great question. I know it's similar to your work as well. I think that I guess looking at least in the myocardium, the resident macrophages have been shown to be more efficient at efferocytosis, and so I think and like specifically looking at my data with the large peritoneal macrophages that express TIMD4 and MERTK, I would probably suspect that those are the macrophages that are the first ones to eat, as they already have all the machinery in place to perform efferocytosis. And so, yeah, I think that is one of the probably biggest confounding variables within the pseudotime trajectory. In that, so I do think the large peritoneal macrophage population is probably the first one eating.
Thank you.
Okay, great. Okay, I think that's all the questions for now. So I'll hand it over to Lale to introduce our next speaker.
Okay, great. So our next speaker is Laurent Noir-Chauvet. He is a faculty, a director of research at the University of Nice. And his lab is studying the metabolism of immune and hematopoietic systems in cancer, myeloproliferative disease, and cardiometabolic diseases. And Laurent has already shared his screen. Okay, Laurent, go for it.
And thanks, Katie, Esther, and Abru for the kind invitation to present some of the data we have and the new data. And I'm very happy to present with Ed. I think it's thanks to Lali, probably, who managed to put the Columbia people together. So the title of the presentation will be the metabolic reprogramming of macrophages in atherosclerosis, and is it all about cholesterol? So we know that lipid modulation will not meet the patient need for cardiovascular risk reduction, and every day we have more than 50,000 patients that are dying from cardiovascular disease. I don't want to make a comparison with COVID, but we could. Despite the fact that we have statin or other lipid lowering therapy, you can clearly see that there is still a need for novel therapeutic approach. And if you look at the paper from Paul Ridker a few years ago, clearly what he showed that the residual inflammatory risk is very high, more than 29% in patients taking statin. And if you add on top of that, the dual cholesterol and inflammatory risk, you can go up to 45 to up to 50%. And it is even more in the general population if you look at the paper. So what is exactly inflammation? Inflammation is usually based on the CRP when you have a level of CRP higher than 2 milligrams per liter in the blood. But it's not that simple. I mean, we all know that there is a change in the inflammatory cytokine in the blood of the patient. There is also immune cell alteration. And you also have a local or tissue inflammatory response depending on the disease, which is most of the time associated with an immune cell infiltration. And as Connor and Ed introduced today, the tissue damage and cellular death are probably a key event in the inflammatory response of the tissue. So the origin of the inflammation is still not completely clear. We know that both innate and adaptive immunity are contributing to the inflammatory response. But if you look at the different signaling pathway, it starts to become very messy, and we still don't know what is really the origin of the inflammation. At least if you look at just one signaling pathway, the interleukin-1 beta pathway, from the contostrial, you can see that targeting the interleukin-1 beta with this blocking antibody can really achieve a low level of inflammation, and that is beneficial for cardiovascular outcomes. So this was the first proof of concept that targeting the inflammatory pathway was crucial. But what are the novel approach to inhibit inflammation? We know from the group of Matthias and Phil Swirsky that stress is probably one of the key components contributing to enhance the myelopoiesis or the hematopoietic system, acting through the brain in part by different hormones that are secreted. We also know that the environment is very crucial. It could train immunity. We know that bacterial infection, probiotic, and vaccine could contribute to change the disease. But what we know less is how energy contribute to all these different mechanistic steps, not only changing the bone marrow hematopoietic system or locally affecting the macrophage behavior. So what we have found when I was at Columbia is that cholesterol is really an integral player in the immune response in atherosclerosis, working both at the level of the bone marrow and the hematopoietic stem cell, changing the expansion and proliferation and pushing the system toward monocyte production. And this contributes to an increase in monocyte infiltration into the plaque, where macrophages become more inflamed, and we have described a lot of different mechanisms in macrophages that contribute to this inflammatory response. But cholesterol is only a small part of the iceberg, and we know that immunometabolism is already an emerging field. The field was started in the early 50s by Otto Warburg and Hans Adolf Krebs. But really, the term of immunometabolism become very familiar for all of us in the early 20s, which is the key metabolic pathway to immune cell development, fate, and behavior. And Ed and Connor has spoke a lot already on all this recent literature that has been described on macrophages and fatty acid oxidation and mitochondria. So what I would like to do today is try to divide the presentation in three steps. The first step, so basically the three most important nutrients, which are sugar, fatty acid, and amino acid. And I will start with the glycolytic control of low-grade inflammation. So I heard that Ed Fisher was present today. So we know from different groups, including Ed, that hyperglycemia is really linked to myelopoiesis and cardiovascular disease in preclinical model. And Ira Goldberg was also part of all this study. And we have recently reviewed with Bertrand Cariou all the data that are known in the literature and in patients looking at lowering hyperglycemia in type 2 diabetic patients and how it affects the inflammatory response. So this is in type 2 diabetes, and this is just an example here with the SGLT2 inhibitor, which has shown a very potent effect on decreasing cardiovascular death events. And so there is more data coming that should come soon from the BRIT, showing that there is a big effect on the metabolism and the hematopoiesis in human, probably in part by controlling the fate of this hematopoietic stem cell. But today I will focus, I will just discuss about non-diabetic patients. So we know from FDG PET CT imaging using an analog of glucose, which is the 18F FDG, that you can predict the secondary cardiovascular events in the large cohort like the Framingham study. And so what has been very interesting over the last few years is that in part the group of Tawakol and Matthias Narendorf have shown that if you look in patients with acute coronary syndrome, so you know that you have more glucose uptake at the level of the atherosclerotic plaque. But what was very interesting is that in fact it's correlating very well with the amount of glucose that are taken up not only by the bone marrow, but also by the spleen. And if you look at the correlation, it's very linear, suggesting that it's a general change in the glucose utilization and the general inflammatory response that is observed in patients with cardiovascular disease. So we know that GLUT1 is one of the key transporters in the hematopoietic system. We know that it transports glucose into the cell, where it will feed the TCA cycle and provide energy, not only to maintain intermediate for anabolic creation, but it's also a producer of very large amount of ATP, a large amount, not 36 to 48 ATP, but very quickly. And so we decided to test the hypothesis that GLUT1 could be crucial for atherosclerosis development, and to prove the concept, we backcrossed the ApoE knockout mice to a GLUT1 haploinsufficient mice, and we look at atherosclerosis in these mice. We first found that there was a 35% decrease in glucose uptake, not only in the bone marrow, but also in the spleen and in the aorta. And this was associated with a decrease in monocytosis and a decrease in atherosclerosis in these mice, suggesting that in the mouse model that there was a causal link between glucose utilization in the hematopoietic system and the atherosclerosis development. So we decided with Phil Swirsky to summarize all the literature that was known about the change in glucose or cholesterol metabolism at the level of this hematopoietic system, driving the myelopoiesis, and this is summarized in this review in 2018. And basically all the risk factors that are known to affect atherosclerosis are also controlling the metabolism of this hematopoietic stem cell. So that's for the glucose. What about the dyslipidemia? It is well known that there is an imbalance between the pro-atherogenic TG ApoB containing lipoprotein and the anti-atherogenic ApoA1 lipoprotein in cardiovascular disease. And we have tried to review all the recent literature on the new drug in the post-statin era to manage atherosclerosis. And as you can see, you have lipid-lowering therapy that are shown here, but you have also a lot of new therapy that are targeting the chylomicron or the VLDL by targeting the cholesterol reabsorption in the intestine or the lipolysis of this lipoprotein. I would like to focus for like 1 second on this BPA, because BPA has been developed to inhibit the cholesterol synthesis. It could be an alternative to statin. And in fact, BPA is also an inhibitor of the first step of the fatty acid synthesis. So it could have a dual effect, not only by inhibiting the cholesterol synthesis, but also by inhibiting the fatty acid synthesis. And it has recently emerged as an alternative therapeutic approach in patients with cardiovascular disease. And in fact, we have the first evidence that in macrophages in atherosclerotic plaque, we look at the regulation of all these fatty acid synthesis genes. They are almost all upregulating, the one in red here. And we know that change in the fatty acid in macrophages lead to a lot of change in inflammatory response. The group of Ira Tabas and Oliver Soloin have shown that the lipoxin resolvin are very important as inflammatory mediator. We also know that the PUFA are very crucial to change the epigenetic of the cell, but also the activation of the nuclear receptor. And there is more and more evidence that the balance between saturated fatty acid and polyunsaturated fatty acid can change the TLR4 signaling pathway, for example. So we have started to use the BPA into macrophages and start to see that you have a strong decrease in the inflammatory response. And I won't show you the data, but basically, if we look back in the literature at the different model that targets the different enzyme at the level of the hematopoietic system, you can see that there is a consensus that all these different knockout models limit the development of atherosclerosis, suggesting that this fatty acid synthesis pathway is very crucial in the inflammatory response. We were also interested in this other enzyme called LIPA, which is a lysosomal acid lipase. And it's interesting because this lipase has a dual effect, not only in hydrolyzing cholesterol, but also in hydrolyzing triglyceride. And it has been shown in a GWAS study that SNPs in LIPA was associated with cardiovascular disease. And probably in this study, LIPA activity is downregulated. So it's a central player in lipid handling. And so we decided to try to understand what could be regulating LIPA activity. And we tried different settings with macrophages by loading the cell with different bacteria, lipids, oxidized phospholipids. And what we found basically in this study is that LIPA activity is best regulated when the efferocytic index change. So basically, more you have efferocytosis, the more you have LIPA activity. And Connor discussed a little bit about that in this presentation. And so what we have decided to do is to see if LIPA was important during efferocytosis. So we perform both in vitro and in vivo experiments. And what you can see, if you load an apoptotic cell, which is here in blue, and we fed, and this cell were loaded with BODP, which is a cholesterol analog. Basically, what you can see is that in a control cell, the cholesterol is diffuse because LIPA is active. But when we block LIPA with a ladlistat, which is a specific inhibitor, you can clearly see that the cholesterol is stuck outside of the cell all around the apoptotic cell, and it's not going into the macrophages. So we look at whether these changes was associated with change in membrane ruffling. And so we look at a double staining for RAC1 and F-actin. And as you can see, in the control cell, RAC1 and F-actin, the cytoskeleton is going around the apoptotic cell to allow the uptake and digestion of the cell. But in presence of the ladlistat, you can clearly see that there is no cytoskeleton rearrangement and the cells are stuck outside of the macrophages. So in vivo, when we injected apoptotic cell by an IV injection into the mouse, and we treat the mice with the ladlistat again, you can clearly see that there is much less of this apoptotic cell here in red, in the red pep macrophage of the spleen compared to the control. So suggesting that there is also a defect in efferocytosis in vivo. And the consequence of that, if you look at the spleen, is that the spleen starts to become very infiltrated with yelloid cells, which are very foamy and full of lipids. So the idea is that LIPA could support post-efferocytosis digestion for efficient macrophage repair function. And that's the mechanism are described in this paper from Manon. We found two main mechanisms, one changing the transcription factor for elixir, so the amount of ligand that are generated by the cell. And the second one is a change in the calcium from the mitochondria, which will change the membrane ruffling. And this is a mechanism that has been also proposed by Ira Tabas to be crucial during efferocytosis. So I discussed about sugar, fat. What about amino acid, which is also part of the nutrients, one of the three nutrients in our life. So we were interested in glutamine, which is a semi-essential amino acid. And glutamine represents up to 60% of the body total capacity of amino acid. And you can find it in eggs or steaks, but we were interested in this advertising online on Google is that glutamine could have anti-inflammatory effects. It could improve metabolism. It could have boost your brain function. But in fact, when you look on PubMed, you clearly have no strong evidence or causal proof that glutamine is doing all these things. So we decided to try to understand how glutamine could be important in atherosclerosis. And so what we did is we use a similar approach than the 18F-FDG PET scan imaging, except that we used the 14C glutamine, and that we injected that in the ApoE mice, so an atherosclerotic mouse model. So what you can see here is that clearly you have more glutamine uptake in the aorta on the ApoE knockout mice compared to an healthy control. But very surprisingly, when we run a TLC, which is a very old-fashioned technique, and you look at the ratio between glutamine to glutamate, which is converted by this enzyme called GLS1, you can clearly see that there is no conversion at all to the glutamate, suggesting that glutamine is stuck into the aorta and cannot be converted. And this is probably because GLS1 activity is downregulated into the plaque. So as Connor showed you, we also use the tool from Max Artyomov, which is a topological analysis called COMBT1 topological analysis, or Shiny game also. And basically, you can feed any kind of data set on your bioinformatic tool, and you will get a map of all the genes that are up or down regulated. And we also develop an independent tool, which we call DreamBio, which will feed the same data set directly on the cake pathway. So there is less of a bias by doing that, because basically you will really have the consensus map of what we know in terms of metabolism. So this is basically a kind of result we obtain. We look at the RNA-seq from an ApoE knockout aorta compared to a control after 6 weeks of a high-fat diet. As you expect, you will see a change in carbohydrates. I should discuss about the sugar at the beginning of this presentation. You also see change in lipids. But what was very interesting for us is that this module, which contains all these different amino acids, and especially we found the glutamine module with GLS1 being upregulated in both visualization. So we decided to generate the lysan-3 and GLS1 flux mice and backcross them to the ApoE knockout mice. And what we found after feeding the mice for 12 weeks on a high-fat diet is that clearly these mice develop a strong atherosclerosis compared to control mice. And this has been seen by NFAS staining and also directly on the aortic section. And the quantification of the plaque is shown here on the right. So clearly you have an increase in atherosclerosis development in this animal model. So we also decided to look at the relevance of this data in human. And so we collaborate with Judith Slimmer and Eric Bisson. And basically, we look at the expression of GLS1 in stable human plaque versus unstable human plaque. You can clearly see that GLS1 is downregulated in this unstable plaque. And when you look at the correlation with various markers that they have in their study, you can clearly see that GLS1 is inversely correlated to the plaque size, positively associated with a M2 marker. But what was very interesting for us is that it's inversely correlated to the core plaque ratio, suggesting that there is more necrotic core in human plaque. So we came back to the mouse model we developed, the ApoE knockout mice backcrossed with the lysan-3 GLS1 flux mice. And clearly what we found is that in these mice, in the atherosclerotic plaque of these mice, we have more tunnel cell per section, as shown here. And this is associated with a decrease in efferocytosis, as estimated by the ratio versus tunnel positive nuclei versus tunnel negative nuclei, which is a technique developed by Ed Thorpe and Ira Tabas, and I forgot, Ed will let me later on to tell me who developed this assay. So clearly what we found is more necrotic core in this atherosclerotic plaque of these mice and less efferocytosis. And just briefly, I won't go into all the metabolic detail. I think Ed and Collard did a great job by showing all how the metabolism control efferocytosis. What we have just shown is that basically glutamine analysis is essential to support membrane ruffling and allow cell engulfment. And so this is electron microscopy showing less ruffling all around the macrophages compared to a control cell. We also did a double staining with F18 and the apoptotic cell are in green. You can clearly see that in control cell, the apoptotic cell are engulfed and in the knockout macrophages for GLS1, you have basically no engulfment. And we also developed this assay from the Tabas lab, showing that basically the macrophages with GLS1 deficiency, so less glutaminolysis, have less unsealed macrophage apoptotic cell after second efferocytosis. So just briefly, if we have just to have a one take-home message, immunometabolism will become a new actor for future cardiovascular therapy. It's very crucial. And I try to summarize here all the literature from the different groups in the field, especially in terms of efferocytosis and how glucose, lipid, or glutamine all together contribute to change the mitochondria phenotype, will change the transcriptional reprogramming of the cell, and of course, the electron transportation will have many impacts on the NAD interleukin 10 pathway from head work, and also on the calcium and membrane ruffling to allow the cell to take up macrophages. So with that, I would like to thank all the people involved in this study, my lab, of course, in Nice. We also have advertising on our website for new postdoc position, and of course all the funding agencies that are supporting this one. Thank you again for everybody.
All right, Laurent, it was a great talk. So please raise your hands if you have a question. We don't have the chat function. Just raise your hand, and I will be able to unmute you. So while we're waiting for questions, let me ask the first question. So Laurent, what do you think? How do you think GLS is regulated? So what is the upstream? What are the upstream factors you think that regulates GLS?
So we still don't know. We have been looking for a long time at epigenetic changes in the promoter of GLS1. There was nothing very special about that. There is few transcriptional factor that has been identified, but we haven't looked at the activity into the atherosclerotic plaque yet to see the relevance in vivo of the finding we found here. So I don't have a clear answer in vivo, at least for you.
Okay, let me ask another question. So I think it was Mori Birnbaum's group, right? They also showed a similar type of regulation at the hepatocyte. So do you think there's like a common metabolic regulation of glutamine degradation, you think, at the macrophage and hepatocyte?
Yeah, one of the big difference between macrophages and the hepatocyte and the paper of Birnbaum is beautiful, but the isoform that are very highly present and quite specific to the liver is GLS2, not GLS1.
Right.
GLS2 is barely expressed in macrophages, so they are very, very different regulation of both enzymes. We still don't know why GLS2 is so specific of the liver, but that's probably very different mechanism. So we are working with, on GLS2. So if you're interested, I'm sure you will have a lot of feedback for me on that. So.
Okay, wonderful. So we have two questions. First, Trevor? Oh, yes, Trevor, I unmuted you.
Can you hear me, Laurent?
Hi, Trevor.
Hi, I have a question for you. So in your GLS1 knockout macrophages, do you think that they're eating the dead macrophages differently, or do you think GLS1 might be killing the macrophages in a different manner, which is, maybe through necrosis or some other cell death, so they're not getting eaten properly?
I'm not surprised you are asking me the question, Trevor. No, we have tried all the different death pathway. We were very interested in the ferropoptosis because glutamine has been involved in this specific kind of apoptosis. we can reproduce the paper on ferropoptosis with GLS1 under very specific conditions as they have done in their paper. But under our condition, with just lipid loading or just an inflammatory insult, there is no death in our GLS1 knockout cell. So basically, it's very specific on the membrane ruffling, and we have all the data showing that under this specific setting, there is no change in death, any kind of death.
Great, thanks.
Okay, and then we have another question. Ada. Okay, Ada, I unmuted you. Okay, we cannot hear you, Ada.
Okay,
yes.
Can you hear me now?
Yes.
Yes.
Thank you. How are you? I said it was a wonderful talk, as always. So I was wondering if GLS1, you showed some human data and there was some connection with arginase 1. And I was wondering if you looked at multiple efferocytosis events, if it regulates the multiple efferocytosis events. And for both LIPA and GLS1, if there's any role in hematopoietic progenitors.
So the first question is that we try a lot of different settings for efferocytosis. One thing which is very important, and I didn't show here, is that we know from a science paper 2 years ago that when you prime the macrophages with interleukin-4, you will boost the efferocytic response. So we saw the best change with GLS1 deficiency under this setting, but we have also tried the second efferocytosis protocol from Tabas, and under this setting, you have also a defect in efferocytosis. So there is not only one mechanism by which GLS1 control the efferocytic response. There are probably several ones, and it's always in all the different settings, the GLS1 play a very important role. So that's the first question. The second was the hematopoietic system. To my knowledge, LIPA is not expressed in the hematopoietic progenitor, at least from all the database that you can find online. We never tried to see, we know that there is some kind of leukocytosis into the knockout mice that has been reported in the past. We still don't know if it's secondary to the huge inflammatory response due to the macrophage, the defect in macrophage efferocytosis, or if there is something else into these mice. But I don't think anybody has looked at LIPA in the hematopoietic progenitor, to my knowledge. So that's the first. GLS1, we are working on the role of GLS1 in the hematopoietic progenitor. So there are some effects in these mice, but we had to develop another mouse model to look specifically at this pathway. Lies and pre are not a good model. Thank you.
Okay, and we have another question, Marit.
Okay, yeah, thank you. Laurent, great presentation. So I just wanted to get back to one of the first experiments you performed with the radioactive glutamine, where you basically saw no conversion at all in the aorta into the glutamate. So then you say that was because of very low GLS1 activity. Do you have any idea how that would be, what would be the reason of that low activity, why you would observe that?
That comes back to the question of Lali on what regulates GLS1. So we have some in vitro evidence of some different pathway, but in vivo we need to test it. I mean, we have no clue in vivo on why and how GLS1 is then regulated. That's a very good question, but I mean that's part of the next, I guess.
Could it have any thing to do with the plaques, for instance, being very advanced in the ApoE knockout, because you see that's like low expression in the unstable plaques in humans?
That could be, but that won't tell you the mechanism.
No. Yeah, but still.
We do think it's very important for the composition of the plaque, and I mean I didn't show all the data that we have on composition, but there is some changes in the plaque size and in the plaque composition. but the human data really points to the major defect being the necrotic core.
Yeah, sure. Okay, thank you. Thanks.
Okay, great. Thanks, Laurent, again. Thank you, Connor and Ed as well. We thank our speakers, and we hope to see you next week. Ebru, would you like to say, would you like to have?
Yeah,
Yes,
thank you so much for joining us and to our speakers. Great talks. We'll see you next week.
See you next week, everybody.
Thank
Goodbye.
you very much.
Bye.
See you all. Bye-bye. Take care.